program, and also particularly from the minor track, and one of the major actually. My name is Daniel van den Hoven. I'm assistant professor here at Maastricht University as well as in Germany, in Würzburg. And today I was asked to give you a lecture on the role of epigenetic regulation in well, psychiatric disorders. I think in the title or in the course description it says depression, but I took it a bit broader because I don't think this is covered anywhere else in the program so far. Let's first make it clear that you can interrupt me when you think it's needed. I mean, the learning goals of this, the things you need to recall, do not cover the whole presentation, so it's particularly to get you enthusiastic about this line of research and to give you some insight into where the field is at this moment. Because, of course, you particularly know epigenetic regulation in light of the regulation of gene expression, and particularly in relation maybe to cancer, for example, as a possible disorder where epigenetic dysregulation has major implications. Nevertheless, evidence is emerging that it also plays a very important role in relation to psychiatric disorders. So again, feel free to interrupt me when needed, and also when the pace is when it's going too fast or too slow. Just let me know what you know already. Yeah, because this differs from year to year. Anyway, to start with, maybe a background on the work I'm involved in. Can you still hear me like this in the back? Yes? Maybe when I get tired towards the end, I speak up less loud, so warn me. Let's dim the light a bit. Don't fall asleep, please, but... Stage, I need less light on the stage. Alright, so this is sort of an overview of the work I'm involved in. What we actually look at is how gene-environment interactions shape the phenotype, and in this case, the behavioral phenotype in relation to psychiatric disorders. And we look at it from a lifetime perspective, meaning that we already study risk factors, environmental exposures, as well as protective factors from early life onwards, actually from conception onwards, until actually aging and related disorders like Alzheimer's disease. So we look at things from a lifespan perspective. We look at genetic variation because we know that certain genetic variants make you more or less susceptible to develop a psychiatric disorder. So there's also a lot of genetics involved. And these are some models, some genes that we are particularly using. And here you might recognize those in light of Alzheimer's disease. I will get back to that later. So, for example, I did my PhD on developmental stress exposure, stress during pregnancy, and how this affects the offspring, so not the mother itself, the pregnant woman, but the offspring in terms of their risk of developing psychiatric disorders later in life. We also look at adolescents a lot. Maybe an appealing example is cannabis exposure. You may have used it or may still use it. You may have used it during adolescence, like I did. Nobody told me it was wrong. Actually, it's one of the major risk factors to develop psychosis, particularly during this phase of life, because the brain is susceptible for the influences, the detrimental influences of cannabis exposure. And it's one of the major environmental risk factors of developing psychotic disorders during that phase of life. We also look at others, social defeat, so how do you deal with social stress when your peers look down on you. Stress in general, we also look at CO2 exposure as a model for panic. We look at inflammation, which is known to also increase the risk of developing, for example, depression. But we also look at positive influences like certain compounds that you may use, but also caloric restriction. We'll get back to that later. Then we look at this in light of certain psychiatric disorders. This is all done in close collaboration with Feyerdahl or the psychiatric hospital here in Maastricht. And we don't just look at vulnerability, but also at resilience. And what do I mean with that? I mean with that, okay, everybody in the past had classical research approaches. Okay, we compare patients to controls, and we then look at genetic differences, I mean epigenetic differences that discriminate between the two. Nowadays, people are more interested, okay, we know that the whole bunch of people exposed to chronic stress, and why does one person develop depression and the other one not? We're not just interested in the ones that do develop, but particularly in the ones that do not develop a disorder. Because maybe in their brain there is sort of a key towards how you can increase resilience and how you can increase coping with stress and how you can at least design also pharmacological treatment strategies based on those protective mechanisms that somehow are present in the brains of these subjects that are resilient. So not just looking at vulnerability, but also the results. And then of course eventually we look at a different set of big set of outcome measures. Me personally, I always try to connect epigenetic processes to behavioral phenotypes. So I want to see to which extent these epigenetic changes contribute to differences in behavior related to emotional regulation, for example. And in the back you may see the corpse of a rodent with the skull of a human, meaning that we always, in every PhD project that we have up and running, we try to do the clinical studies as well as experimental work, either in animals or using in vitro models. So we always try to make a translational step, because if I look at postmortem brain tissue, you're not sure whether the epigenetic patterns are well conserved, you have to control for this. You can't directly, in living patients of course, access the brain, so for that reason you can switch to animal models. So there's always an intricate crosstalk between various models that we use. Overview of the dog, brief intro. I don't think you need a lot of background information on epigenetics because you, if correct, have already been introduced to that. Yes? Okay, I'm going to go quick. I will still show some methods to study epigenetics, particularly because in psychiatry you have limited access to tissue. So techniques are sometimes slightly different, or at least you have access to a limited, to a subset only of those techniques. I mean, in cancer you can at least investigate the tumor, in a brain disease you can only do that when the person has disease, which is a bit. Some study examples, I think these are particularly the nice things. We'll look at some examples of how epigenetics may be involved in psychiatric disorder, and then some discussion included. I think some of you may already have the PDF. I just added a few slides because we have some recent human data added to that. It's just a few slides. So again, interrupt me where needed. We can skip this, I think. Chromatin modifications and DNA methylation. These are the two we're particularly interested in, but also in linears. I don't know whether microRNA regulation has been discussed also. Yes, but maybe more on that at the very end. What is important to mention, you also know this probably, is that of course epigenetic processes are the key factor of explaining why one cell develops into a neuron and the other cell into a blood cell. This has been explained as well? More or less? No? But, I mean, this cell, there's only one cell with one set of DNA. Two cells with an identical genetic makeup, of course. Then why eventually do we, at a certain cell stage, I think it's a 16 cell stage in an embryo, why do we see differences emerge? Well, that's actually because the environment for those different cell types is different. And from that moment onwards, epigenetics starts to kick in. Let's say we have this stage, where we have two cell types and they still have an identical environment. They have an outer environment which is identical, and they have a shared environment on the inside which is identical. In the 4-cell stage, still the same. But from the moment onwards, that some cells lie on the inner mass of your embryo and others on the outer mass, you get a difference in the environment. Some of them are only surrounded by other cells, whereas others are surrounded by other cells as well as by the external environment. So from that stage onwards, you have a different environment for every individual cell type. And this is the trigger for starting differentiation. Because now we have a different environment which has a different impact on the genome, in this case on the epigenome, causing differences there epigenetically and eventually mediating differential gene expression and therefore phenotypes of the cell. So that's the basic mechanism of epigenetics in a nutshell. Or at least this shows the implication. Of course, you can also look at this at a more, let's say, sophisticated scale, and then eventually you can see that this results in neurons versus blood cells or even different types of neurons. It's just a matter of a different environment, and eventually, of course, cells at the start in some stages will already have a different epigenome. But from the very first start, they all have the same. So along similar lines, as I said before, you can actually also then, even if you have two identical neurons, if they are exposed to different environmental circumstances, maybe in one case to low levels of high oxygen versus normal levels, they can develop differentially. Of course, there might also be already epigenetic differences in those cells, causing them to respond differently to a similar environmental variation. Actually, you could see epigenetics as a sort of cellular memory, because the memory of what this cell has encountered in the past is more or less stored in this epigenotype, and therefore you can predict how this cell will respond to future environmental variation. So you could see epigenetics as a sort of cellular memory. Interestingly, if we look at twins, who are supposed to be genetically identical, they are not. Why not? So let's say we have identical twins. Why are they not genetically identical? I'm not talking about epigenetics, but about genetics. Do you know that every generation there's more or less 100 or more de novo mutations? So even when we look at animal models which have been crossbred for more than 20 generations, so brothers and sisters have been crossbred, rats and mice often, before they are used in the lab. So we say, okay, they are genetically identical. And what we then try to do is we expose them to different environments, stress versus control, for example, and we say, okay, anything that you see in terms of the phenotype is independent from genetics because they are genetically identical. They are not. In every generation there are more or less 100 de novo mutations, so also in identical twins. So they are not completely identical. But let's assume that influence of the genetic background is negligible because it's a random process, and you never know whether such a mutation directly affects the phenotype. Let's assume they are genetically identical. They will eventually develop differently based on epigenetic mechanisms, because in the womb they have a different environment. One may have a better blood supply than the other, and they are competing actually for resources. So they are competitors. One will get bigger than the other. That's what you almost always see. So we have actually a different programming of this epigenome and of their physiology, based on the difference in the womb. And actually, it is well known that the lightest one at birth has an increased risk of developing all kinds of disorders. And this ranges from cardiovascular disorders, to diabetes, insulin resistance, to depression. So the lower your weight at birth, the higher the risk of developing eventually depression or schizophrenia. The higher the risk, don't panic. Yes. It's just like with smoking and getting lung cancer. Anyway, so even genetically identical twins, they have different disease susceptibility, which starts already early in life and which is mediated by epigenetics, and eventually influencing the physiology, etc. The interesting thing, of course, is that we can more or less reverse, or at least, since the environment can impact on the epigenetic machinery, the environment can also reverse some of these changes eventually. And this can also be done by treatment. And what is now actually done, they are testing some of these H-tag inhibitors. You know what H-tag inhibitors are? So you have heard of histone deacetylation. So let me go briefly get to that. So these are the two basic epigenetic mechanisms. Here you see differences in methylation, for example, at a single CpG level, which have been explained. Then we have these chromatin modifications. We have positive modifications that generally open up chromatin state and allow the transcription machinery to do its work. And acetylation, histone acetylation is one of the best examples. There's also histone deacetylation, which then normally repress gene expression. And then we have these compounds that are called histone deacetylase inhibitors. We say H-tag inhibitors or hashtag inhibitors. They are often used in cancer therapies. But they are also being tested in psychiatric disorders. Actually, it has been discovered that something like valproic acid, which is given to people with bipolar disorder, for example, acts on the epigenome and is an H-tag inhibitor. So actually compounds that have already been out there for a while, they show to influence directly the epigenetic phenotype and thereby maybe also causing cancer. So you can also reverse some of the symptoms maybe. We're talking about behavior and psychiatry via treatment. Of course, you always have to consider what are these side effects. This will still take a few years before we know all we can. Of course, you can imagine that maybe the risk of developing cancer will increase. Okay, well, nevertheless, in general, you probably also know this epigenetic variation adds another level of regulation on top of genetic variation. And interestingly, because I was talking already about early development, in this case we're actually already influencing 3 generations at the same time. We are influencing the mother, in this case you see smoking, which we mostly use the example of stress exposure during pregnancy. You're influencing the fetus, and in case of female fetus, you might know that the egg cells are produced early in life, creating the life. So actually in the embryo or in the fetus, the egg cells for the next generation are already being produced. So different from sperm cells. But you're already influencing 3 generations. This of course is all makes sense. This is more, let's say, direct effect. I will also show you later on an indirect effect of transgenerational inheritance of epigenetic traits that may influence how you behave, so you can trace this back to your grandmother. YDNA is your destiny, big paper in Times Magazine, and actually it is still your destiny. The only thing is that the epigenetic processes in DNA are more important than initially thought, and it's not genetic variation as such that is only important. So it actually is still your destiny. Just the modifications that have been added to it. And this is then the concept of what we do. We look at genetic variation, only occasionally, in psychiatry at least, a certain genetic variation directly causes a disease. For example, Rett syndrome is caused by mutation in one single gene. It's the MECP2 gene. Oh, what a coincidence, it's a gene that regulates epigenetic regulation. But okay, only coincidentally, you have a mutation in a gene that causes a disease. At least in psychiatry, yeah? Cancer does. In most cases, we see the environment having an impact directly on the phenotype. So, for example, stress is a well-known risk factor of develop psychiatric disorders. But nevertheless, in most cases, we see interactions, so that your genetic backgrounds makes you more or less vulnerable, and also then they actually need this environmental exposure in addition to the genetic variation. And then in between, we have the epigenetype that regulates gene expression and eventually the phenotype. Yeah? So this is the concept. We can skip that. So some methods. You've probably seen all these, except for immunohistochemistry. Or didn't you discuss yet the methods? Just simply not yet. No? Okay. We'll briefly touch upon a few. This is actually work that we did in our own group. It's very cheap because you might know that studying genetic and epigenetic variation is quite expensive. If you want to run some micro experiment, and in this case a micro experiment based on immunopresentation of, for example, methylation, that's quite costly. For a simple experiment, you already pay 20,000 to 30,000 euros. What you can do is also look at epigenetic mechanisms using immunohistochemistry. You just use antibodies against layers that are involved in epigenetic regulation. So, for example, in this case, an antibody against a certain DNA methyltransferase. So that's an enzyme that mediates DNA methylation. I'll show a bit more on that later. And then you can compare non-demented subjects with Alzheimer's disease subjects. Again, more on that later. So it's a very simple technique, and this will cost you 500 euros to compare 2 groups, and you have a nice paper. And you know more, of course, in terms of the truth. Chromatin immunopresentation. So you probably have heard of this. Use an antibody against a certain chromatin modification, and you can use this to immunoprecipitate parts of the DNA which have then been more or less affected by this chromatin modification. And if you then combine this with an array approach, so that on the array you want to see which genes are differentially acylated or methylated, at least at the histone level, then we speak of chip-on-chip. Still, it's already quite expensive. Of course, more sophisticated methods use chip sequencing, and they are even more expensive. So what you use, for example, this is an example where you use transcription factors to identify, or to do exactly the same with chromatin modifications. Anything that binds to the DNA in terms of protein, you make the DNA, make small pieces out of it, but you first cross-link all these proteins with the DNA so that they don't let go. And then eventually you use your antibody of interest and you immunoprecipitate your regions that have this specific chromatin modification, and then you determine them, for example, by an array experiment or by any other means. You sequence the pieces of DNA so you can start to see, okay, the patients have more acetylation at this gene when compared to controls. And if you combine this with an array, you speak of chip on chip or chip. You can do the same for methylation, and in this case it's much more, it gives you much more information, or at least the outcome may be more important. You have a antibody against methylation, so you identify all the spots in the DNA where you see more or less methylation between your two phenotypes or the two groups of interest. Why would this be easier, or why would I be more enthusiastic about mated when compared to chip? Just guess. I don't know the names anyway, so you can just guess. How many chromatin modifications do exist? Speaking of one chromatin modification that is interesting too, or you should know the answer to this question. Just guess.
Over 1,000. Over 1,000.
Yes, it's numerous. It's maybe even you can't count, because it's a combination of these different chromatin modifications that determine eventually whether a gene is being expressed more or less. Okay, there are a few that are more interesting than others. How many methylation patterns exist? How many different degrees of methylation exist? Well, methylation is actually one. Okay, we also have hydroxymethylation, and we'll get back to that later, but you actually identify any piece of the DNA of your genome where you have differential methylation. So to study the whole genome in terms of methylation, you need one experiment which is comparable to one single chromatin modification. And if you want to have more insight into chromatin modification, so you would need thousands of these experiments to get as much info. It's not black as white as I now say, so, but at least in methylation you can do more for the same amount of money and for the same material. Blah, blah. Yeah. What you use is an antibody against methylated cytosine. They are quite specific, and then you immunoprecipitate all the pieces where you have this methylated cytosine, and then you sequence them, for example, or you can use an array approach, and you look between your two conditions. Okay, which gene is more or less methylated in this condition? And of course, you can go with a high resolution at whatever you want, depending on the available resource. Let's do this. Well, eventually, what all is important then, particularly also in psychiatry, is a combination of genetics and epigenetics. So let's assume that we have a certain locus of interest, and we have a SNP at this spot that is known to associate with a certain phenotype. In that same gene, we also have epigenetic variation. So either this C can be methylated or not. We speak of certain haplotypes because in this case we always see that this T and G, they segregate together. So you actually can focus on one of them, then you already should be able to follow the stories. So let's assume you have a T and a G. This means you actually have no phenotype, so you are normal. Let's assume you have a G and A. It's always annoying, blue on black, okay. Just remember blue. You have some phenotype. From the epigenetic point of view, this is all genetics. From the epigenetic point of view, you can either have methylation or not. Let's say if you have methylation, you have no phenotype. If you have no methylation, you do have some phenotype. Then there's always a combination between the two that eventually is interesting. And let's say you have both the vulnerable epigenotype here and the vulnerable genotype here, you have a certain phenotype. This is eventually where you have to go. You have to combine genetics with epigenetics, but of course, no psychiatric disorder is a single gene disorder. At least, there's only a few. So that makes a difference. Some study examples, and this is much more interesting. I just wanted to give you the technique so that you don't all of a sudden have slides that I showed in the chemistry lecture. We'll talk about sophisticated mechanisms like epigenetics. Again, this is going to use this line of research, and I will first show you some examples that are related to prenatal stress and early life development, and later on also some examples of aging. So you might have heard of this word, epigenetic regulation of the glucose receptor. So what can we see? These people were interested in the relationship between childhood-induced suicide and differences in HPA axis. So the HPA axis is the hormonal stress response system. So if you have to do some public speaking and you're not used to that, so it might be that they want to explain something later on, then you get a stress response, first sympathetic regulation, fight or flight, but in a while you also get stress hormones that reach your peripheral organs, etc., and have all kinds of effects. You probably are not hungry, etc., and some functions are suppressed and others are still not. Anyway, so we have changes in the stress response system, which happen associated with suicide as well. So childhood abuse increases the risk of getting suicidal, and also changes in the HPA axis that are associated with suicide and this support. Well, this is the HPA axis. You can have a look at it later. What is important for now is that HPA axis has a sort of feedback. So let's say the stress is gone, then eventually this response should be terminated, and this is happening via glucocorticoid receptors, particularly at the level of the hippocampus. It has been shown that people with a depression sometimes show less of these glucocorticoid receptors, so something is wrong with their feedback. So they are continuously stressed even when there's no real reason for it. And if they are really stressed by a trigger, then they can't shut down this response properly, which eventually causes all kinds of damage also to the hippocampus and to other stress systems. Yeah? What did these people show here in 2009? They compared control subjects with suicide subjects. So this is in the brain of non-abused people and people that were abused during early life. So this was documented for these people. And what they saw is that the expression at the mRNA level of this glucocorticoid receptor was reduced in those that committed suicide and were exposed to early life childhood maltreatment and abuse. Which was not the case in the ones that also committed suicide or were not abused. So this shows that early life experiences, in this case, had a long-term effect on the expression of this gene, which is known to be important in stress regulation. This is more of the same, and here they then show that this effect was mediated via an increase in methylation at the same gene. So the effect on expression is mediated via DNA methylation. So what is the problem with this study? What is lacking? Well, my name should be on here, but that's not the thing that's lacking. Which group would you like to have seen to be added?
Non-suicidal, but maybe abused.
Yeah. So the controls are not divided into abuse or not. So these controls died with a natural cause or an accident or just for any other reason. And so of course we don't see any effect whether this effect of childhood abuse is also present in the controls. Next to that, all these groups committed suicide. So what's the big deal? So what is then the role of this glucocorticoid receptor if they all committed suicide? But nevertheless, it was one of the first papers in humans showing that epigenetic processes can affect the brain and can have a long-term effect on brain function, because we know for sure that glucocorticoid receptor expression is important in regulating adult stress responsivity. Nevertheless, still some flaws in the stuff. But of course, such brain material is sparse and it's already very nice. So this is the name of the gene. There was a lower expression and no effect of the ones that committed suicide without being abused. Something similar was done in animals, and there you can go much more in depth. So what they did here, in this case in rats, they let me first show the normal mechanism that has been discovered. It's very important how the mother takes care of the pups. So the father is not involved. You see that more often in some humans, of course. But the mother takes care of the pups. She licks them, feeds them, grooms them, etc. And the levels of grooming appear to be important for how this animal, the pup, behaves in adulthood. So the better you are groomed, so let's say we're dealing with normal levels of maternal care, by a certain mechanism, this eventually affects the expression of the glucocorticoid receptor. So you have this sort of normal regulation of the glucocorticoid receptor, which is regulated only during the first week of life. So this is P1, 1 day after delivery, after birth. Then there is an active demethylation of this glucocorticoid receptor gene. And this only takes place during the first week of life. So if you change levels of maternal care later on, you don't see an effect. So we have a demethylation of the promoter region of this glucocorticoid receptor, which eventually gives you a normal expression and also then results in a normal regulation of this hormonal stress system. Now, if we have low levels of maternal care, We can mimic this by either using animals that, based on their genetic background, show low levels of maternal care. We can also stress the mother so that they show low levels of maternal care. We can take the mother away from the pups. We can do this in many ways. And what you see then is that this demethylation doesn't take place as efficiently, resulting in less glucocorticoid receptor expression, and you see also a variable phenotype. So this fits more or less what you saw in the human. They don't commit suicide, but you see some other changes in behavior that fit with that. What you can now do then is influence this via pharmacological intervention or by any other means related to food intake, for example. So in this case, so this is what I more or less explained at the beginning, you can have from a close inactive compact DNA, you can escalate the chromatin and make gene expression possible. There's also the reverse pattern, using in this one deacetylation, HDAC, and what I said, there are HDAC inhibitors, in this case PSA, which then inhibit this compaction of DNA of chromatin. So you would expect the DNA to stay more open or to become more open. And that's what they did here. So later on in life, so not during this first week, we start off with low levels, so we have a reduced expression of the glucocorticoid receptor. And later in life, in adulthood, they give them this HDAC inhibitor. And they are able to reverse epigenetic patterns and reverse the effect on the expression, eventually increasing the expression of the glucocorticoid receptor and resulting in normal behavior again. So it's very nice that you are able to influence this even after the damage more or less has been done. And that's the nice thing about epigenetic versus genetics. They also did something else. They also influenced action on the methylation level directly by giving a methyl donor. So normally if you give more methyl donors, you would get more methylation, so more compact DNA and less expression. It's more or less a rule, but it's not always the case. More methylation does not always mean less expression. Okay, let's assume it does. What they did here, we have high levels of maternal care, so normal phenotype. All of a sudden we give more methyl donors in adulthood, and what we see again is that now in this case glucocorticoid receptor expression goes down, and we get an animal that shows more anxiety and depressive-like behavior. So we are also able to change the phenotype into the wrong direction, so to say. And this mimics actually the animals that have low levels of maternal care, although now we have high levels. This is more or less clear. These results have not been replicated for years, because this was actually already from, I think, 2005 or so. Yes, it was. Yeah, 2005 or 2006. And they now just discovered that this was not methylation purely, but hydroxymethylation. I will get back to that. It's not crucially important, but interesting. Okay, here you see the label results more or less summarized. You see the glucocorticoid receptor expression. You see if they have low levels of maternal care, they normally have a low level of expression. If you then increase acetylation via PSA, this HDAC inhibitor, you are able to increase the expression again to a normal level. These are, let's say, the control animals who receive high levels of maternal care. If you then give them this methyl donor, as shown over here, their expression goes down. And similarly, you see corresponding changes in the phenotype, although it's not completely the same, but let's ignore that for now. That was the first example. I hope that was clear. I think so. Now it's getting more complicated. Because I actually can't explain it. Well, I can explain it, but I don't go after this. I told you this more or less direct effect of transgenerational development. With direct, I mean that a certain effect, a certain environmental component, can immediately affect one generation, two generations, and three generations. More or less direct. It takes a while before you see that, of course, in the third generation. This is something you can grasp. What these people did, and I will try to explain the design of the study. Since you have the slide, or you can at least print them out later, you can have a more closer look at these, which should eventually work. You might be able to follow it. I hope I can explain it already clearly now. Okay, what happened here? What they did here is we have control animals and maternal separation animals. So again, this means that they take the mother away from the litter. So they take the mother away from the pups. Yeah, that's the basic start of the experiment. If you do this for a short while, this has actually a positive effect because the mother comes back and then takes care of the pups extra, actually. If you do this for a few hours in a day, this has a negative effect because the mother is really stressed. Yeah? In this case, they did it for a long period of time. So, believe me, the mother is stressed, she gets back to the litter, and this affects not only the phenotype of the mother, but also of the pups. Yeah? This is something we can all follow. Now, what they did, they first of all studied, of course, the offspring, they had one generation, but later on, they also took females of those pups, took females and bred them, sorry, I'm saying wrong, they took males and bred them for 1 day with a naive female. So they took a male from this first generation and then put it with a female so that she gets fertilized, but then they take the male away. Why is that important? If you see any effect in the next generation, it's not caused by an indirect effect of the behavior of the father or something like that. The father is just with the mother for 1 day, it's enough, or 5 minutes might be enough, but so anything that is transmitted to the next generation must be transmitted by the sperm cell. Yeah? They do the same with the generation thereafter, so I'm going to show you some behavioral data of that F2 generation. Then they did the same with the next generation. They took a male of that second generation, put it with a female for a day, and then looked at the next generation in the F2. Yeah? That's the design of the study. I have it in small with the big picture might be difficult. Anyway, what they did, they looked at the mothers and saw that the mothers showed differences in maternal behavior. So the mothers that were taken away from the pups showed lower levels of maternal care. So that fits the previous study that I showed. What's particularly interesting is that they that the effect of maternal care was not seen in the models of the next generation. So in the next generation, as I said, they just took a male from this first generation and bred it with a female, naive female that was completely new, and then looked at the offspring, etc. And of course, because this female has not been influenced by anything, she shows normal maternal care. Yeah? It's just to show that the effects in the following generations are not mediated by this naive mother. Yeah? So we have a phenotype in the mother that was stressed initially, and what we see here, this is F1 generation, and it doesn't matter what type of behavior is shown here, but I can tell you that these male pups, the male offspring, show more depressive-like behavior. And that's actually shown over here. Let me see. Time spent floating in the, no, this is F1 generation. Time spent floating. So this is a certain behavioral test, a forced swim test we call this. An animal is plunged into a beaker with water, and what is known is that animals normally show motivation to escape. However, if they are stressed or show depressive-like phenotype, they don't show these attempts to escape that often. So they are less motivated to escape. Certain test for behavior, for depression. Anyway, what you see is that the males, and only the males of this first generation, show more depressive-like behavior. So they show this behavioral phenotype. And their mother actually showed lower levels of maternal care towards these spots. This is the F2 generation. So again, we took those males, bred them with naive females for a day. We didn't do that, but others. Bred them with a female for a day, and then look at the next generation. And there actually we see an effect in the female. So in the next generation, we don't see anything in the male anymore, but in the female. In the following generation, so we take the males that are not affected here, we take them, we crossbreed them with new naive females. And they are not affected. And what happens in the next generation? The males are affected. Yeah, this has not been replicated yet, as you can imagine, but more and more evidence is pointing in this direction, that there's transgenerational inheritance of behavioral traits via the germline. This can either be the maternal germline or the paternal germline. And there is no possibility of any indirect effect of the paternal phenotype. Right? The father is not involved in the upbringing of the animals. There's no paternal care here. So the effect has to be by the germline. I don't know whether you know this, but until 4 or 5 years ago, everybody said in the sperm, methylation patterns are completely erased. And you don't see anything of that back in the next generation. Well, now it has been shown that this is not the case. And that's what they showed here. So forget all these numbers and so on. What they show here is that there are certain genes which show similar methylation patterns in the brain and the sperm. So the brains of these animals and the sperm of these animals. And that there are differences between your stress animals versus your non-stress animals, and that this is transmitted by the germline. And that some of these patterns are comparable in the brain and the sperm. So So that's amazing. Why would this take place? What would be the rationale behind it?
You can provide better survival for the offspring.
Yeah, that's actually the point. You pay close attention in the vegetable. So, for example, prenatal stress or developmental stress. Imagine a case where you have high predator density. So where a prey that is pregnant and there's high predator density. So there's a lot of stress. And at that time, it makes sense that you program your offspring to be more anxious so that they are alert and escape these predators more easily. So what happens is you get a programming of the HPA axis and a whole kind of other mechanisms. The physiology is adapted to survive under such circumstances. So that's actually what is taking place. So you actually adapt your physiology based on the environmental expectations of the environment. So actually what you do is you have your whole genotype, your genome, which provides a huge window of opportunity, and you narrow this down based on the actual environmental circumstances. This is shown at many levels. If you are born in a country where it's quite humid and hot, you will develop more sweat glands when compared to when you were born in a region that is quite dry. Things like this is all epigenetically mediated, independent of genetic background. Of course, you can get the problem of a mismatch. If you end up in an environment that is non-stressful, you might get problems. For example, what you see also with these low birth weight babies, so stress during pregnancy also results in reduction of birth weight. This also means that they expect low nutrient availability after birth. They expect low levels of food being available. So what they do is they take up food as efficient as possible. And that's why low birth weight babies get obese that easily. So they get bigger later in life because they store food much more easily. This is mediated by epigenetics. The problem is, if we have a low birth weight baby due to stress instead of due to low food availability, and we then end up in a society with McDonald's and Burger King, is that there's a mismatch between what was predicted during pregnancy and what is eventually really going on. And this mismatch causes disease. Of course, in animals we can expect these stressful mechanisms, stress-related mechanisms, to be important for programming the brain. But if you then end up in a society, or in a human actually, where natural selection doesn't work like this, then you get a complete mismatch ID, and you get an increased susceptibility to disease. All right. That was understandable?
I have a question. Do they also say how it that is sex dependent, because the first generation is male, second female, and the third is male again?
This depends largely on the genetic background. So this is one animal strain. There's one strain of mice, I think, in this case. What we see, we do a lot of prenatal stress experiments. We've done that with different strains of mice, with different rat strains. You might know Spigoli rats, Fisher rats, Lewis rats. And it depends largely on the genetic background of that strain. So for example, if we use Spigoli rats, then we see that male offspring is more affected in response to prenatal stress when compared to the female. If we use mice, in most cases the females are more susceptible. So this depends on genetic background. What is known in general that you might know that psychiatric disorders in most cases are more readily seen in women versus men. For example, depression is I think the ratio is 2 or 3 against 1, showing more chances of developing depression in females than women. You don't see that clearly when you look at developmental stress exposure. So stress during early development may cause different subtypes of depression, which are also readily seen in males. Why then do you see the switch between generations? Nobody knows. The question is, if you use another strain of animals, will you also see this switch? It has been reported before, but these are things we can't grasp. That's what I mean. I can describe this. These are the data, but why this takes place in such a manner, I don't know. Why you have programming, there's a clear reason for that. Makes sense. Why this skips a generation, because that's what happened. The first generation was a male, and the second generation a female, and then again a male. No. Of course, in humans we can't study it. In transgenerational, then you need a study of 150 years. So it's pretty complicated. But it might be that if you use different genetic background of the strain used, especially on animals, that you see something. You can imagine that something happens indirectly also with the programming again of the X cell and X cell. But one generation, at least the rest of the cells.
Whatever happens to the mother and then the baby inside and the eggshells, wouldn't have made more sense for them to test maybe females?
Yeah. So the question is, why did they test the males and not the females? Well, the test with the females, I didn't show that, but these were more classical approaches. So that has already been done. So of course, in most cases where you affect the female offspring, you also affect the next generation, because if, let's say, if the female offspring gets pregnant herself, she also shows difference in behavior that again affect by maternal programming, also by maternal care, the next generation. But that is often, you then can't discriminate between a direct effect and an indirect effect. What I mean is, some of the effects might be mediated by programming already the X cells when being a fetus, but it has been shown that some of these effects are mediated by maternal care. There are experiments that try to control for this using cross-fostering. So they then take a stressed mother or a mother of which the mother was stressed, and then switch her pups to an unstressed mother and vice versa. And then they show indeed that these transgenerational patterns are always seen when dealing with females. So this was another level where we don't have this indirect effect of maternal care, and where we do this effect of programming of the Excel already during yeah, and these traits are then submitted to the next generation in a sex-dependent manner. And what I showed in the last two slides was that you see methylation patterns being associated with this difference in phenotype, and you see that both in the brain as well as in the sperm cells. Nevertheless, they only took a few genes. It was a candidate-based approach, so you don't know to which extent this takes place. What shall we do? Shall we have a small break, short break, and then continue 10 minutes, or do you want to continue? Break. Break? Yes, go on.